Hi guys, thanks for tuning in. So this is going to be the first in a sequence of videos recapping some of the questions from the recent mock exam. So I'm going to start with the biology paper and eukaryotic and prokaryotic cells with a bonus round of magnification. Okay, so life can be broadly broken down into two different groups in terms of their cellular structure. The first being eukaryotic. So this incorporates protists, animals, plants, and fungus. The other direction we have prokaryotic, which is basically all of your bacteria. So in your exam, you're most likely to be questioned on this in the form of identifying cell structures, substructures, are they prokaryotic, eukaryotic, and so on and so forth. So in the recent mock exam, you were presented, in higher tier paper anyway, you were presented with 4 cells, A, B, C, and D, with various structures, organelles, and so on and so forth. So which cell is a plant cell? With one reason. So it's 2 marks. It'll be 1 mark for identifying which cell and 1 mark for stating your reason. Your work on plant and animal cells should cause you to move towards cell D as being the obvious answer for question 1. There are a host of reasons for this. First of all, you've got the vacuole, the sap-filled vacuole. You've also got the chloroplasts, which are these structures here, which contain the chlorophyll, which allows the plants to photosynthesize. This is actually a palisade mesophyll cell. You can tell from the long rectangular structure. These sit just below the surface of the leaf and allow the plant to photosynthesize. So a good answer would state D. Reason: it has a vacuole. Careful if you're spelling a vacuole, V-A-C-O-U-L-E. It is also perfectly acceptable to say it contains chloroplasts. In the second part, you're asked which is the animal cell and give the reason. So again, it's worth 2 marks. We're going to say 2 separate things here. And if you have a closer look at the 4 types of cell, there is one obvious choice, and that is B. The reason for that, it's the only one that does not have a cell wall. You can see that cell A has got this thick cell wall here, so has cell C, and so has cell D. Animal cells do not have a cell wall. So a good answer would be B. No cell wall. I saw an answer recently that said it only has a cell membrane. Now, it may have a cell membrane, but it doesn't only have a cell membrane. So this is an example of an answer that sounds good, but actually is wrong. It has cytoplasm, the stuff that fills the cell, where the reactions are carried out, and it also has a nucleus. So you can't say it only has a cell membrane. Yeah, I know what you're trying to get at, but you have to explicitly say that there's no cell wall. So do be careful about how you're wording your answers. You were then asked to state which cell was a prokaryotic cell and give a reason. So your prokaryotic cell is your bacterial cell. Now, there are a couple of ways in which you can distinguish which cell is your bacterial cell. The first thing to look out for is, does it have a nucleus? Prokaryotic cells do not have membrane-bound DNA. They have plasmids, these loops of DNA, which are essentially loose in the cytoplasm. So immediately with that knowledge, we can determine that C has to be the prokaryotic cell. No nucleus. Or DNA not bound by a membrane. Okay, finally, there was a question on magnification. You're given the magnification, so remember this is how many times bigger the object will appear, so what we call the image size, okay, compared to the real size. And you're actually given the equation in the question here. So the size of the image is 25 millimeters. So that is this top bit, so 25 millimeters. We're told that the magnification is 100,000, so that's going to be equal to that over whatever the real size is. So we've taken the numbers from the question and we've put them into the equation there. And it's just a case of cross multiplying that correctly. So essentially these two are going to swap over. So the real size will be 25 millimeters over 100,000. Now if we put that into a calculator, we get 0.00025 millimeters. However, we need the answer in micrometers, and this will be quite a common theme for a microscopy question where you're dealing with very, very small objects, incredibly small. So how many millimeters do we have in 1 micrometer? Well, you have 1,000. Oh, sorry, there we go. 1,000 millimeters is equal to 1 micrometer. So the symbol mu, this U with like this front tail, micrometer, micro. It's 1,000 times smaller than a millimeter. So therefore, our decimal point here is going to move 1, 2, 3 places to give us 0.25 micrometers for our size. Okay, well I hope you found this video useful. I'll just provide a little summary. Recognise where our eukaryote cells are and the key features. Recognise the prokaryote cell, the bacterial cell. So DNA not contained within the membranes, usually a bit of a giveaway. Sometimes we see a flagellum, the tail, whip-like structure helps it to move, but look out for those key features, know those key features. Make sure that you are stating very explicit, clear reasons with no room for mistake, no vague answers. And when you're dealing with microscopy questions, make sure that you clearly substitute in your numbers like any other calculation, particularly in physics. Substitute your numbers into the equation, and ultimately remember your conversions from micrometers to millimeters. It's not beyond possibility that they might throw in other prefixes there, centimeters and so on and so forth, so you need to be able to comfortably move between them, so get practicing. If you've got any specific requests in terms of what type of videos you'd like to see on the channel, just drop a comment below. Thank you very much, take care, adios.